Hello, in today's class, we're going to be talking about an introduction to Chip-seq metaplots. We'll do this in two parts. First, we'll go through conceptually how to create a metaplot and what a metaplot is in this presentation. And then we'll go to RStudio and actually create some metaplots using the data that we've run through Nextflow. So a metaplot is really a way of asking what is happening in your sequencing experiment genome-wide. And it doesn't have to be just with Chip-seq. It can be made with ATAC-seq or DNA hypersensitivity data, all sorts of sequencing data. So in a way, a metaplot is an averageogram of whatever your sequencing signals are relative to some genomic annotation. So I'm going to show you an example now for enhancers. Here we see in the center of this is the center of each enhancer in the genome lined up, and then the total signal for DNA hypersensitivity here in the y-axis. You'll often see these accompanied by a heat map where each enhancer in the genome is lined up as a row in the heat map, and the color is the signal in that enhancer. So just to show you that these can be used not just for enhancers, here's an example of a gene body metaplot where each gene is scaled so that the transcription start site and the transcription end site line up, and then you can visualize here GC content across the genome-wide. Similarly, this can be done for promoters, and that's what we'll be doing today. It should be noted that a metaplot can represent the sum of whatever sequencing signal or peak occurrence, and today we'll be using the peaks to create the metaplot. And we'll be using pol2 as an example. So we expect pol2 to be bound near promoters, and here we see a bigwig track visualized in UCSC for pol-R2b. That's the second largest subunit in pol2. And you can see that the peaks highlighted here in red are pretty much lining up with promoters as we would expect. So the first step conceptually is that we'll just define the promoter regions. So in our case, we use 3 kb upstream and downstream of the transcription start site. So here are some promoter regions here. And then really what we're going to do is just take and line those regions up so that the TSS falls in the same place and make a summary across all of those promoters to make this metaplot. Here's some examples of metaplots from last year's class data. And you can see that there is quite a lot of variation in how binding happens that metaplots can highlight. So some DNA binding proteins like ATF2 have a very tight window around the TSS where they bind. Others bind a bit wider around the promoter, and others are actually depleted right at the transcription start site. So the data that we'll need to start building our metaplots is our Chip-seq peaks G-ranges object, which basically consists of all the peaks called from the Max software, and also a gene annotation G-ranges object. And we'll be using GenCode annotation for this. So our first step is that we're going to transform the gene annotation into promoter windows, plus and minus 3 kb around the transcription start site. And the code that we'll be using to do that looks like this. The main function that we'll be using to do this is called promoters. It's a convenience function provided by genomic ranges that takes into account the strandedness of the gene so that you can have asymmetrical upstream and downstream windows. In our case, though, we'll just use 3 kb both upstream and downstream. Okay, so now we have these two objects, the promoter G-ranges object and the Chip-seq peaks G-ranges object. And the second step that we'll need to do is transform the peaks G-ranges object into a coverage object. What that will do is place a 1 where peaks are and a 0 where there are no peaks in the genome. So the result is a long vector that's approximately the length of the genome of zeros and ones. So if we stored this vector for the whole genome, which is 3.2 billion base pairs for the human genome, it would require about 150 gigabytes of memory. So we need to compress this somehow. And you'll notice that there are long runs of zeros followed by runs of ones. So what we can do is store just every time a 1 changes to a 0, we can store that and say that there's 1 1 followed by 20 zeros followed by 4 ones. etc. So this is what run length encoding is. And it's very efficient because it turns what would be a 150 gigabyte vector into a few kilobytes. The code that we need to run this is very simple. We'll just use the coverage function from genomic ranges and apply it to our peaks object. Okay, so now that we've got our coverage object, when we get into the code, you'll notice that we do a number of housekeeping steps at this point. And primarily the purpose of this is that the peaks may not extend to the end of each chromosome. In fact, it's unlikely that there would be a binding event near the end of a chromosome. So there may be some promoters outside the range of our run length encoded vector, and we need to filter those out before we move on. So as an example here, this promoter on the very right, we would be removing. Okay, after that, we just have this coverage object and our promoter granges object. And the third step is that we'll subset this coverage vector to just the places where it overlaps with the promoter windows. So we end up with just the information corresponding to where this coverage vector overlaps the promoters. And for that, we use a simple command also from granges called views. Okay, now the fourth step is that we'll construct a matrix of the coverage values for each promoter region. So we're just lining up those coverage values and making them into a matrix. So with that, we'll turn our run length encoding vectors back into vectors. That's highlighted in red there. And then we'll just call an rbind on that object so that we bind the rows into a matrix. After that, you'll notice that we have both positive and negative strand promoters. And since in our final metaplot, we want all of the upstream binding to be to the left of the plot and all of the downstream binding to be to the right, we need to flip those around so that they're all aligned in the same direction. So we'll just flip the negative strand promoters and their corresponding coverage vectors, and then we have it all lined up. And the code to do that is here. Highlighted in red is the portion that will flip the coverage vectors so that the last column in the coverage vector is first and the first column is last. Finally, we just need to sum up the columns, normalize it, and then make the plot. So this is what it will look like. And the code to do that is just call sums, and then our normalization will just be dividing by the sum across that vector so that the vector will sum to 1. And here's the resulting plot. So even though in our example we used polar2b, we'll use polar2a when we move to RStudio. Okay, so I've pulled up our Metaplots R markdown here, and I'm just going to walk you through it. So at the top, we load in the libraries that we need and also our functions in order to import the peaks and some plotting themes. We'll set a path for where the data exists and then just import our GenCode annotations along with the consensus peaks. Okay, so you'll see here that we have loaded in the environment the consensus peaks list, which is the peaks from all of our DNA binding proteins in the study, and also a GenCode granges object. So if you recall, the first step is to create promoter windows from that GenCode gr object. And we'll do that first by filtering the GenCode object down to just genes, since there are also exons and transcript entries in there. Then we'll use this promoters function to give us 3 KB upstream and downstream of each transcription start site. So now that we've created this object, we know that the width of each promoter should be 6 KB. We can check on that by running with all promoters gr, and we see that we have 6,000. Use table. You can see that we have 6,000 for every entry. Step 2 is that we're going to transform these peaks into a coverage object. So we'll just run this coverage function. And now if we look at this in the console, we can see that there is an integer run length encoding with 862 runs, which means that there was 861 peaks on chromosome 1. and then there is an entry for each chromosome. Okay, so now on to the housekeeping step to make sure that there aren't promoters that fall outside of our peak coverage vectors. So we can use this function to check the length of each coverage vector. And this will give us the length of each coverage vector for each chromosome. Then we'll create a granges object that starts at the beginning of each chromosome. So it starts at 1 and goes only until the end, the length of the coverage vector. So we're now creating a granges object which has one entry for each chromosome, and the range is the length of the coverage vector. Then we can use subset by overlaps to subset our promoters to just those that are within this coverage granges object. So one more housekeeping step is that we want to make sure that we have the same chromosomes for our peak coverage vector and for our promoter vector. There could be some contigs listed in the promoters object that don't exist in our peaks object. Or there could be some binding proteins that are very sparse binders and don't bind on some chromosomes altogether. So for that, we'll take the intersect of the chromosomes in our peak coverage vector and the chromosomes from our promoters. So that will be our chromosomes object, and we can see that we have 24 in common. Then we'll subset the peak coverage vector by this, and subset the promoters object by this. And that way we also ensure that they're in the same order for the functions we have moving forward. Okay, so the third step is that we're going to subset the peak coverage vector to just the portions overlapping the promoter windows. And the views function will do that for us. Views will take a coverage vector and subset to whichever windows you supply as the second argument. In this case, it's our promoter windows. So once we run this, we can check it out, and we'll notice that it's a list where each chromosome is an entry, and then within that, there are views, which are 6,000 in length, and it's basically showing us the values of the coverage vector for this promoter here. Next, we'll construct a matrix of the coverage values for each promoter region. So in order to do that, we will be walking through this promoter peak view object that we just created and change each entry to a vector. So what we're going to do is apply as.vector to each element in this views object using view apply. And this is an anonymous function here that we've set up to do that. And lastly, the final thing to note here is that we will transpose the vectors so that they're arranged column-wise, so that in the matrix, each column will correspond to a position within our promoter, 1 through 6,000. So after we run that, we just need to convert it to a matrix where we can use rbind to row bind into a matrix. So you'll notice now that this has become a much larger object. It's 1.3 gigabytes in size. That's okay to do now since we've just taken the subset of the coverage vector that's over those windows. But as I said previously, if we converted from integer ranges to vectors, when we had all the data, it would be about 150 gigabytes of memory. So we're nearly there. The next step is to align the positive and negative strand promoters, and we're going to do this by flipping the values of the negative strand. First, we will retrieve which promoters are on the minus strand and store the index into this value here. So this is telling us that the second, third, and sixth promoters are minus strand. Then we'll use this to index to only the rows of our new matrix that we created and flip around the columns so that the last column comes first and the first column comes last. So this will give us 6,000 through 1 in reverse order. So there are now a lot of promoters in this matrix that might not have any binding events on them, and they're uninformative for our plot. So at this point, we can filter those out by using row sums and say we want to take only those rows where the row sums are greater than 1. So there's at least one base pair covered by a peak in that promoter. That reduces the size of this matrix a lot. It is now 129 megabytes versus 1.3 gigabytes. So finally, we can sum the columns of this matrix using col sums, and then we'll divide by the sum of this vector that we've created, which will give us the sort of density where the overall vector will sum to 1. This gives us the peak frequency, or where the peaks are occurring in this vector. In order to visualize it using ggplot, we'll create a data frame with an x value. So the x value will range from -3000 to 3000. And then we will have this vector that we've created, which is the normalized column sums, as the second column in this data frame. So that data frame looks like this. And finally, we can just plot it. So that looks as expected. Pol2 is bound in the center of promoters, but it has a pretty wide profile compared to some other DNA binding proteins. So this plot is basic. Let's add some titles and labels to it, along with a really nifty feature in ggplot is that you can actually put text as a label on your axis. So we will just call the 0 point the TSS. Great, so now we've made our first metaplot. Of course, we'll be using this quite a lot, and that means that we can put all the commands that we just ran into a function so we can use it later conveniently. So all the steps that we just took are here in this function. And at the very end, it returns a metaplot data frame, which is equivalent to the data frame that we just created. So this is a little long for a function, but it does accomplish one specific task, and that means it makes sense to put it into a function together. Lastly, just to illustrate using this function, we can make separate plots for lincRNA and mRNA, or rather separate profiles for lincRNA and mRNA, and then we can plot them together to see if Pol2 is binding differently to lincRNA or mRNA. So in order to do that, we'll take this genes object that we created earlier from the GenCode annotation, and we'll filter so that we only get the gene type, which is equal to lincRNA. So now that we've got that, we can see what the length of this is. And it's 16,000. So in this version of GenCode, there are almost 17,000 lincRNAs. We'll use the same promoters function to grab 3kb upstream and downstream of each of those genes. And then finally, we'll just give it to that function that we just created and let it return a data frame of our metaplot values. We can now just do the same for mRNA genes. The GenCode term for mRNA is protein coding. Okay, so now that we have a data frame for the lincRNA metaplot data and the mRNA metaplot data, we can combine them into the same data frame so that we can plot them in the same plot. In order to do that, we'll create a new column in each data frame called gene type, and this column will indicate whether that data point corresponds to mRNA or lincRNA. So for the mRNA metaplot profile data frame, we will make that column's value all mRNA. And for the lincRNA, we'll make the gene type column all lincRNA. So now, since these two data frames have the same column names, we can bind the rows, which stacks up the data frames end to end, and then we have a combined metaplot profile data frame. With that, we can plot this, and the only new parameter in this plot from the last one is that we have a new aesthetic called gene type, and this will plot a unique color for each unique value in the gene type column. So here we have two values, so we'll have two colors with one color assigned to mRNA and the other assigned to lincRNA. So we'll plot this. The other plot function here to note is that we scaled the colors manually because these colors look nicer. So what we can see from this is that pol2 binds more tightly to the promoter for mRNA and is a little bit more dispersed over lincRNA promoters. So that's all we have for today's class. We'll be getting more familiar with the metaplots throughout the class.